Great. Well, it's great to be back with some good friends and meet my old colleagues again. That's fantastic. Eva sort of kind of set the scene quite well here. A lot of interesting comments and challenges. So I don't know if I can live up to that, but let's see. And he's also given me an interesting title here about what can and can't be done with modern bioscience and why it's important for Africa. But I need to talk about that without getting too technical, which is a bit tricky, actually, you know, because we've got quite a few technical people in the room, and I don't want to sound like a complete idiot, but at the same time, I don't want to talk too much technology, and the people who aren't that much into bioscience are going to get a bit confused. So, you know, if I get things wrong, or if I get the angle wrong, you know, stop me and shout. Anyway, let's just sort of start off with setting the scene a bit, because, you know, to follow up with what Eva was saying, basically, I think we all know that we had the first green revolution, and if we just look at cereal yields only, in Western Europe, we had this huge increase in cereal yields, which is this blue line on the graph. And it's leveled off quite a bit in recent years, but as you could see, sort of through the Green Revolution, there was this huge increase in yields. But, you know, really there was no change in the area that was harvested down here. If you look at Africa, there was no change in yield, because that's down here, but there was a huge increase in the area that was harvested. That's this one up here. So Africa was increasing its production, but it wasn't doing it through any increase in yield through the Green Revolution. It was just putting more and more land into cultivation. And clearly, you know, that's not something that we can carry on doing forever and ever. There has to be, you know, a whole change in how we handle agriculture in Africa. But even in Europe, I think we can say that, you know, although there was a green revolution, and you know, although it wasn't really seen in Africa, because it wasn't focused on Africa's needs, and yet even in Europe, there were a whole lot of downsides in the sense that it was associated with environmental degradation and was increased use of agrochemicals and irrigation water. And so if we're going to move on from here, you know, what can we do for Africa? Well, can Africa leapfrog then to a doubly green revolution, as people are talking about, where we're going to have some sustainable bioeconomy and at the same time as boosting the output? Well, I mean, as Eva said, that's a tall order, because we've got not just to produce more, but we've got to also produce it to use the biomass for a whole lot of different applications as well. And there's no way we can do that without using the modern bioscience to do it. So that's really the challenge that we've got. So what do we need? Well, obviously we need crops with durable disease resistance. We need a whole lot of different abiotic stress tolerances. We need nutrient and water use efficiency. And then we need all those crops for industrial uses, for the biofuels, for the pharmaceuticals, for the green chemicals. And I think what people tend to ignore as well is that we also need to look in the livestock question. There's a huge focus on the crops, but we mustn't ignore the livestock in all of this. Particularly because, you know, as people get a little bit more affluent, they want more meat in their diets, and that's actually causing a huge revolution in agriculture as well, the fact that people want more meat, and that is, you know, processing of grains into meat is not a particularly efficient thing to do. So we get protein, but at a cost. But if we are going to have livestock, at least we need to have reduced greenhouse gas emissions from the livestock, and we need more efficient nutrient conversion. So we need to focus on all of those things. Well, okay, so then I get into a bit of what the bioscience tools are that we've got. And there's a huge sort of array of things that we've got already. What we're doing with them is another question, but let me just sort of take you through a bit of some of the things that we've got in our toolkit already. Well, I mean, obviously one of the most sort of basic things we've got is tissue culture, where we can get disease-free planting material, and that's already being used in Africa, particularly for banana tissue culture, mainly for being used for some of the vegetatively propagated crops. But it's also the basis for all the other technologies that come after. I mean, we wouldn't have the GM crops if we didn't have the tissue culture as the basic tool that we're working with. But there is a whole lot of potential now to use tissue culture techniques to do different things as well. So there's the potential to produce interspecific and intergeneric hybrids with increased vigor. So, you know, I think there's many crops, well, there's some crops where we have hybrids, and maize clearly is a case in point, but I think there's potential to have hybrids from other crops as well, where we could increase the vigor of the crop and get increased yields. And some of those things we can now start to do with tissue culture. There's potential also for hybrids in crops such as banana and cassava, which are the vegetatively propagated crops, which could give us some productivity increases. So some new things that are coming through with tissue culture. I think there's also a potential probably to make more use of tools like mutation breeding that I think we don't do enough with. But then, well, marker-assisted selection, that's something that's been around also for a while, but is coming through more as we move on. Again, it's not a tool that's being used enough in Africa. You know, there are courses run on how to do marker-assisted breeding. A lot of those courses around in various countries in Africa, but I'm not sure that the tools are actually being used a heck of a lot. And that's something that we need to do more of. Because if we can associate a marker with a particular breeding trait, then we can do rapid selection of individual plants or animals, so that we don't actually need to wait for the animal to grow or the plant to grow. We can do it a lot quicker. But even, you know, then beyond sort of the basic things that have been around for a while, now that there is a whole lot of more tools for next generation sequencing, and we can do rapid sequencing of whole genomes, we can do a lot more high throughput genotyping and genome-wide selection. And we've already getting to the point of having whole genome sequences of a number of African crops. So some are including some of these orphan crops. So pigeon pea, teff, which is a big one in Ethiopia, banana, African rice, cassava, those genome sequences are all coming through. And that's going to give us a whole new tool set for things that we can do with those crops. On the livestock side, we also have whole genome sequences of cows, pigs, chickens, sheep, goats. And again, we've got to use that information to put that into the breeding programs. And to make these crops and these animals appropriate for African conditions. I mean, it's no good producing, you know, high-yielding dairy cattle that are fine for the European condition. You put them into an African farming situation, and they're just going to fall flat on their faces. So, you know, we've got to have breeding targets that are appropriate for African conditions. We're also then moving into, you know, a lot of what's resequencing, which is basically looking at variation across the whole genome associated with specific traits. So, you know, you've got your one basic genome sequence, but then you've got to look at, you know, a whole population of plants or animals with different traits, and then try and determine what variation you've got in that genome. So, but when you've got the whole genome sequence, you've then got the potential for simultaneously selecting a whole lot of desirable traits. So it's not just having to look for one marker for one trait, but you can simultaneously look for a whole lot of different things. And you can do that right at the beginning in the breeding program without having to wait for your plant or your animal to reach maturity, as I said. But, however, one of the problems with this is that you then need your breeding populations, because if you don't have a breeding population that reflects all those different desirable traits, you know, particularly for some of these complex characteristics, then you don't have the basis. You don't know what should you sequence. You know, you've got to say, well, here you have all these plants, and you know that this population has got these characteristics, and you can then try and select for those characteristics. But where you've got orphan crops, where there hasn't been a focus on conventional breeding, and you don't have those breeding populations, that can be certainly a bottleneck when you come to doing your genome sequencing. And again, coming back to the livestock, you've got to have those breeding populations for animals that perform well under conditions of limited feed, drought, disease, heat stress, etc. And we don't always have those breeding populations. So we're going to need, you know, there needs to be this interaction between the bioscientists and the conventional breeders to make sure that they really work together. Because, you know, the molecular biologists are great at thinking they've got all the solutions, but they need the people on the ground who can assist them. And I think that's an interaction that we need to build more strongly. Well, then, of course, we come to the whole issue of the GMOs, which is, as Eva has said, is an interesting debate in Europe. Coming from South Africa, you know, they're well entrenched in South Africa, although even there are still, you know, quite a bit of debate about particularly some of the crops that are more relevant for Africa. I mean, it's very easy in South Africa, you know, to deal with an application from Monsanto or Syngenta when they come with, you know, the herbicide tolerance and the insect-resistant maize, because we know how to deal with that. As people like Marietta knows, it's a lot more difficult when you come with a genetically modified pearl millet and what you do with that. And that's when you start to get the regulators kind of a bit hot under the collar and not sure actually what to do with it, and then it kind of goes into a limbo somewhere and nothing much happens. So yeah, we need to move on from those basic traits of the herbicide tolerance and the insect resistance. Nutrient enhancement is certainly coming through. I mean, there's a whole lot of initiatives for, you know, vitamin A and iron and all of those good things, but as yet none of them have seen the final commercialization. I mean, how many years has golden rice been going around the block and back again? And, you know, maybe it'll get through and maybe it won't. But we also are then looking at crops with a whole lot of more complex traits that are in the pipeline. So the drought tolerance, I think, is certainly coming through. At least in the, you know, at least I would say first generation drought tolerant crops are coming through. But I think drought tolerance is not a simple issue, and, you know, although you can get a certain element of drought tolerance with putting, you know, one gene in, how robust that's going to be, I think, remains to be seen. Nutrient use efficiency is certainly going to be an important thing, and, you know, nitrogen efficiency and phosphorus efficiency and all of those things are going to be important, as is stress tolerance. Particularly with climate change, and then pharmaceutical production in plants, which I think we might hear. Are you going to talk about that, Marietta? Yeah. That's, I think, a potential thing for the future, as well as production of a number of other sort of chemicals. But I think, you know, one of the things that we're going to move on from is the whole concept of what is a GMO, what is a GM crop, because of this resistance. You know, that this was the first generation, was the transgenic crops, putting a gene from one organism into a different organism, and people have, you know, a whole lot of issues and problems with that. The question is going to be, for the next generation, where you've got cisgenics or intragenics, where you're transferring DNA between organisms of the same species, does that raise the same set of alarm bells as the transgenic organisms, and that's still a matter of debate in a number of countries as whether a cisgenic would be regulated in the same way as a transgenic. And we don't yet have clarity on that at this point in time, and it may vary from country to country. But it's a debate that I think we as scientists need to engage with the policymakers and the regulators to try and get clarification on this kind of thing. And I think we're now moving into a whole suite of other genetic modifications as well. You know, we've got things like targeted genome editing, where you can do site-specific genetic modifications without necessarily leaving anything in the target organism that originates from genetic modification. So, you know, there's a whole lot of things coming through here, because if you can never in the end identify that you've got any foreign DNA in your target organism, how are you going to regulate it? Because there's no way you can analyze it. You know, you can't prove one way or the other, was this modified or wasn't it? And this goes as well for things like directed mutagenesis of specific genes. There's also this potential using these tools for metabolic engineering of specific pathways, so you can actually modify production of specific end products. You can use this kind of technique as well for eliminating allergens, for increasing disease resistance. And again, as I said, maybe they won't be regulated as GMOs, because a lot of this regulation depends on your ability to actually prove that what you've got there is a GMO. And then I think, you know, coming further down the track, there are things like epigenetic modification, where you're not actually modifying the DNA as such, not in the ACGT sequence, but you are modifying things like DNA methylation states, which can actually be carried through many generations. I mean, people are saying that some of these epigenetic modifications would have the potential to last for maybe 100 years through a number of generations. So that has also got a whole lot of potential for the future, and it's got potential for clonally propagated crops such as banana, where you don't have the sexual propagation, or crops where you've got severe germplasm bottlenecks, so such as groundnuts, where there's very limited diversity within the genome. And you may be able to do a whole lot of new things by epigenetic modification that you can't do with conventional breeding or conventional modification. So that's sort of a few of the things, but then, okay, how's it going to help African agriculture? Well, clearly we need to focus all this research on crops that are important to Africa. We're going to need to focus on climate resistance, and we need to focus on some crops that are relevant for marginal lands as well. Just looking at this, these are some of the crops that are important to Africa. Looking up at the top, it's quite interesting to see that some of the I want to see, where's my thing here? The roots and tubers are at the top here. Actually, I hadn't expected to see that. So there, your roots and tubers, your cereals obviously are next. Cassava is quite a major one here. Okay.
Jane, what's that on the y-axis? Is that tons, million tons?
Megatons.
Megatons.
Yeah.
Okay.
This is off the FAO website. So you can just go onto the FAO website and ask to compare various things, and it shows you all that. Yeah, and then, okay, fruit is next, and vegetables. So fruit and vegetables are here, and then everything else is sort of way down the bottom, which quite surprised me, actually. I thought some of those would be higher up, but they weren't. So we need to be looking at a lot of the root crops. We need to be looking at the cereal crops. We should be looking at fruit and vegetables. Okay? I mean, it's obviously Africa as a whole. I think if you look at individual countries, you're going to get fairly different perspective, but I thought that was quite intriguing. So, well, using all those tools, what can potentially be done right now for Africa? And here I've only talked about the crops. I haven't talked about the livestock, but I think we do maybe separately need to have a debate about crops. But we do need to, we can develop crops with improved nutrient content. That can be done. We can develop first-generation crops with drought tolerance. And we can develop disease-resistant crops. We can develop first-generation crops with enhanced nitrogen use efficiency. And we can develop some improved multi-use biofuel crops. So things like jatropha and sweet sorghum. We can develop crops for production of high-value chemicals and pharmaceuticals. And we can develop oil crops as bio-based alternatives to petrochemical feedstocks. We could use the tools that we've got to do a lot of this stuff. The question is, what actually are we doing? Because I think there's a big gap between what we could do and what we are doing. So the question is, what is in development? Well, these are some of the projects. I mean, clearly I don't know each and every project that's ongoing in Africa, but these are some of the things that stand out. There are projects to develop semi-dwarf and lodging-tolerant teff for Ethiopia. There are projects to develop nutrient-enhanced and disease-resistant cassava and banana. There are projects for biofortified and drought-tolerant maize, for cowpea with disease, insect and striga resistance. There's the orange flesh sweet potato, which is actually already out there, but that's nutrient-enhanced. There's projects for high sugar and biomass sweet sorghum. There are projects for disease and drought-tolerant pearl millet. And there are projects to develop jatropha with reduced seed toxins with the aim that you could then use once you've extracted the oil, you could then use the residue to feed to animals because you wouldn't have the toxins in there that are currently there. So that's some of the projects that are ongoing. But again, even though these are things that are in the lab, there is quite a big gap between having them in the lab and having them out there for the farmers. And this is again one of the major roadblocks, that we don't have that pathway really in place, solidly in place, through the innovation chain to get what's in the lab out to the farmer. And it's a huge problem. But having said, you know, what we can do and what we are doing, the question is then, what can we not yet do but might be on the horizon? Well, there are some important things that need to be done, but where I think we still need to develop the science a bit further before we're going to be able to do them properly. Well, phosphorus uptake is a critical thing. I mean, the world is running out of phosphate that it can use in agriculture. I think many of you know that. And unless we can find better ways to use the phosphorus that we've got, that's going to be a huge limiting factor in agriculture in years to come. For African agriculture, particularly, we need to improve salt tolerance and aluminium tolerance. Aluminium toxicity is quite a big problem in a lot of African countries, and I don't think there's a huge amount being done to address that problem. We also are not yet really able to develop crops that remove toxic chemicals. And I mean, if we just look at some of the mining industries, for instance, and the amount of toxic chemicals that we've got lying around, we could really do a lot more to develop crops that can remove those toxic chemicals, and that would have application in both Europe and Africa. And I think with climate change, I mean, there's a lot of focus on saying, well, we need drought-tolerant crops. But heck, one of the things that's going to be coming through with climate change is we're going to have these huge unpredictable swings. We go from, you know, extremes of heat to cold to flooding, to drought. And it's all very well to plant your drought-tolerant crop, and then you get swamped because you didn't know what was going to come. So, in effect, what we need is crops that are not just drought-tolerant or not just heat-tolerant, but are tolerant to all kinds of extremes that can take whatever nature can throw at them. And that, I think, is going to be a huge challenge. But then, as well, if we're going to develop the bioeconomy, we need to economically produce, and I mean that's economically produce is the key factor here, because it has to be something that you can make a business out of it. We're able to produce a wide range of valuable metabolites in plants, and again, this is something where there's huge potential for Africa, because if you can grow a crop, it's often less skills intensive than actually having a chemicals factory. But there's potential to develop plants that can produce a wide range of metabolites that have got useful products that can go to enhance the economies of the countries. So a wide range of sweeteners, flavors and fragrances, dyes, pigments, skin treatments, cosmeceuticals, nutraceuticals, a whole lot of stuff. But again, there's a lot of talk about it. There's a lot of saying in the science literature, well, this is what we could do, but there's not so much actually happening on the ground. So I would say that's an area for a lot more investment. So then the question is, how can Sweden help? Well, this is just my personal view, but if we look at again, this link between the bioscience and the farming systems, this is where we need action. Okay? We need also, I think, some action here between the farming systems and the products that deliver the improved food security, that mitigate the impact on the environment, that give the new products. But there's a link between the scientists and the farmers that is broken in my mind, but there's also a link between the farmers and the processes in the industry that also needs to happen a lot more effectively, and particularly in Africa. I mean, farmers say, well, what's the point of me producing anymore, because I don't have a market for it. I don't know how to get it to anybody who's actually going to do anything with it. So it's a whole system that we need to build, and we can't just build one component of the system in isolation. We need to build the whole thing. So how can Sweden help to get bioscience to the African farmer for a start? Well, to begin with, we need to enable African scientists to use modern bioscience tools, because in the majority of African countries, people know about some of these tools, but they don't have the funding or the equipment or anything that they can actually use them. And using some of these things is expensive. Just to start using basic marker-assisted breeding, it's not happening because the people don't have the money to do it, they don't have the wherewithal to do it, they don't have the equipment or the facilities to do it. And that all needs to get put in place. And it's no good just putting a piece of equipment in a lab and walking away. It needs a lot more than that. Then there also needs to be support to research programs that really reflect the African priorities. It's no good, as I say, producing things that are going to be like high-yielding crops in an ideal situation, but you put them in the African climate and they're not appropriate. So that, I think, has got to happen. We also need to build more public-private partnerships. The private sector on its own is not going to deliver what Africa needs, but I think there's a willingness amongst the private sector to work with the public sector to help get some of these things on the ground, and the private sector experience can be really valuable. But we can also build public-public partnerships between Europe and Africa, and I think those have got huge value too. So then, as I say, we've got to look at the integrated chain and build those linkages between the scientists, the breeders, the seed sector, which is not developed in much of Africa, the farmers, the industry, and the consumers, so that we've got an integrated value chain. So I think that's really my take-home message, is the value chain approach, that's the way to go. I think that's the end of mine. Yes?